Good morning everyone. Today we are going to see about food microbiology, microbial growth and concerns in various food. So before going into that lecture, the thermal processing of food, though it talks about what are all the thermal processing applicable for the food processing, actually the thermal processing is employed to increase the shelf life of the food products for commercial production, and also it paves the way to have processed food to store it for long time. So it doesn't only depends upon the thermal processing of food, it also involves thermobacteriology and what are all the thermal processing methods available and how to design such process designs and how to validate them in the commercial sterilization or pasteurization method. So everything includes in the thermal processing. So without thermal bacteriology, I cannot design any thermal process to increase the shelf life of the food. So the thermal bacteriology or food microbiology is very much important before we go into detail about various thermal processing methods available for the food processing. So in that concern, in this class we are going to review about various microorganisms available and their concern on various foods and how the food spoilage occurs and everything in bit detail. Okay, so the microorganisms, the moment we say, then it includes bacteria, yeast and molds as well as the viruses. So the most important microorganism of food concern is bacteria. So most of the bacteria are harmless and many are highly beneficial for human beings. So lactobacillus, one such is lactobacillus and streptococcus thermophilus. So it mostly available in the dairy products. Some indicate the probable presence of flith, which is nothing but a transmission of salmonellosis, which is a disease which mainly spreads through flies or cockroaches, and some are the reason for disease causing or spoilage, which is nothing but example is Clostridium perfringens and Bacillus cereus, and few cause disease as well, those were Clostridium botulinum. Okay, so in bacteria there are thousands of species available and they are single celled microorganisms and they are available in the spherical straight rods or spiral rods, and they reproduce by dividing into two cells. Under ideal conditions, the doubling time is about 15 minutes. So each 15 minutes it goes doubling. The rod shaped bacteria exist in two forms. One is the dormant spores and another one is active vegetative cells. Okay, so the vegetative cells form spores under adverse conditions as a means of survival, and spore forms preserve the bacteria from starvation, drying, freezing, chemicals and heat. So these are adverse conditions for the bacterial growth. So what these vegetative cells do is they form spores and keep it as a reserve. When the favorable condition comes, they start forming, becoming a vegetative cell, then it starts its reproduction. So when conditions become favorable, the spores germinate and each spore again become a vegetative cell with the ability to reproduce. So this conversion of spores to vegetative cells and vegetative cells to spores is nothing but a conversion of its form. It is not a pure reproduction. So after forming the vegetative cell only it can reproduce. So most of the sporulating bacteria that grow in the presence of air belongs to the genus Bacillus. Most of the Bacillus bacteria were sporulating bacteria, they grow in the presence of air, and most that grow only in the absence of air is the Clostridium genus. So they are anaerobic bacteria. And one more thing I would like to emphasize here is we are going to see very basics of what are all the microorganisms available and their concern in various food, but I would suggest to you to read more about all these organisms and their concern in various foods with a proper food microbiology book. Okay. So here we are going to only review certain important points which we need to know before designing any thermal processing. So next we will see about the yeast and molds. The example is Saccharomyces cerevisiae and Aspergillus niger. So this is most common, so you might have known already. Are oval shaped or slightly larger than the bacterial cells. They reproduce by budding. So budding, each cell can produce several buds or swellings which break away to form new fully formed daughter cells. So the reproduction happens here by budding. Molds as found on bread, fruit, damp paper or other surfaces are actually composed of millions of microscopic cells which are joined together, form as a chain, and which have numerous branches called hyphae, right. So they form, if you might have seen in the bread surface the black masses or green masses, so which are nothing but a cells joined together to form a chains, and which also have a branches like hyphae. Molds can thrive in conditions too adverse for bacteria or yeast. Okay. So they produce by spores again the molds that are frequently present as a green or black masses on the protruding hyphae. Hyphae is nothing but a branches. So yeast and molds grow on most foods where there are small amounts of nutrient and moisture. It is opposite to bacteria. So the yeast and molds grow on dry fruits, salted fish, bread, pickles, fruits, jams, jellies and similar commodities. So they predominate high salt and sugar environments, whom yeast and molds, but for bacteria they are adverse conditions. Bacteria find conditions of low pH, low moisture or low temperature, and high salt and sugar are unfavorable, but yeast and molds grow in high salt and high sugar conditions. Then comes the viruses. So viruses are smallest and simplest microorganism. Actually they are parasitic and they must need another living organisms to first invade and reproduce. They are called, the living organism with which they invade first is called a host cell, then after that only they can multiply. So viruses which infect bacteria are called bacteriophages. So they cannot infect human beings or other animals. On the other hand, several animal viruses known zoonotes, so can infect human beings. So viruses, they cannot themselves reproduce. So before multiplication they need a host cell. So if those host cells are bacteria, they called bacteriophages. So they won't infect the human beings, but certain animal viruses are there, so they can infect human beings. Actually in the food also it requires a live host cell, without which they cannot multiply in the food. So the infections are two categories. One is the bacterial phage infections. So they are the starter cultures which can interfere seriously with the manufacture of dairy products, which are cheese, buttermilk, sauerkraut, pickles, wine, beer and other desirable fermentative products. They may create an infection. Although viruses require live host cell, they cannot multiply in food. So they can remain viable and infectious for long period of time, and even under highly adverse conditions such as drying, freezing and pasteurization. So even in these conditions they can remain viable and infectious for long time, but they require definitely a host cell, so they cannot do on independently. Okay, so that's all about the basic microorganisms, bacteria, yeast and molds and viruses. So what are all their shapes and which kind of foods they find it favorable conditions for the growth. So then we are going to see what are all the factors affecting growth of microorganisms in the food. Right, there are three ways. One is removing or destroying them by trimming, washing, heating, pickling, by adding chemicals or by encouraging competition between acid and alcohol forming organisms. Actually the first one is, before going for any thermal processing, the pre-treatment would be required. For example, canning operations. So if you want to can any food, so the previous operations were these, I mean trimming, washing, the heating is nothing but they call it as a blanching. So it is done for many reasons. One is to inactivate enzymes as well as to remove the microorganisms. So these to be done prior to food storage or food processing, any food processing. So that way we can reduce the growth of microorganisms, and sometimes it is done by using addition of chemicals, or sometimes we play with the pH so that the competition between the acid and alcohol forming organisms, because we are going to see what are all the favorable pH, temperature and water activity conditions for the growth of microorganisms. So we can control in such a way that so the pH and temperature or water activity is not favorable for the growth of the microorganisms. And some of these pre-treatment methods also we can control the growth of microorganism. And another thing is minimizing contamination from equipment, people and the environment, and from unprocessed food. Right. So, though the main two thermal processing treatments were pasteurization and sterilization, even after sterilization the storage and the handling, for example, we are going for high treatment process. So, there may be, if the equipment is not properly washed or properly sterilized before employing the food for further thermal processing, so that may also contaminate the food. And the handling, the personnel should be aware of the good sanitation practices, and the environment, everything also affect the growth of microorganism. And minimizing microbial growth on equipment by cleaning and sanitizing, and then the product itself by adjusting the storage temperature, pH and other environmental factors. For example, we do a pasteurization process to kill the pathogenic microorganism, but it is the pasteurization temperature is not enough for the it only kill the microorganism, but if it goes to normal room temperature environment, then there may be a contamination of microorganisms again. So, those pasteurized products should be refrigerated for further to increase the shelf life of the food. So, these factors also have to be taken care, because though we process the food by required thermal processing, then further storage, temperature and pH and environmental factors also affect the growth of microorganisms, and sometimes they serve as a favorable conditions for the microorganisms to grow, though it was processed thermally before storage. So, based on that, the bacteria were divided into 4 categories. One is psychrophytes, they can grow only at refrigeration temperatures. The second one is psychrotrophs, they grow well at refrigeration temperature but better at room temperature. And the third category is mesophiles, which grow best or near human body temperature. So, most of the organisms which found in the food is mesophiles, but grow well at room temperature. The fourth category is thermophiles, so which has an optimum temperature of 122 to 150 degree Fahrenheit. The spores can survive around 250 degree Fahrenheit for more than 1 hour. They grow only at the temperatures about as hot as the human hand can endure, and usually not at all or below the body temperature. So, the thermophiles can survive at hot conditions, and it cannot survive below the body temperature, which is nothing but a 98 degree Fahrenheit. Okay, so, there are 4 categories: psychrophytes, which grows at refrigeration; psychrotrophs, which also grow well at refrigeration but better at room temperature; mesophiles, which grow well at room temperature and it can up to the human body temperature, which is nothing but 98 degree Fahrenheit. Thermophiles survive at hot conditions, but they cannot survive at below body temperature. Though we can classify all bacteria in any of the 4 categories, but there are some exceptions. Some psychotrophic microorganisms grow very well, very slowly in foods below freezing, but usually not below 90 degree Fahrenheit. Standard storage temperature for frozen foods are 0 degree Fahrenheit, which does not permit microbial growth. And most psychrotrophs have difficulty growing above 90 degree Fahrenheit. Most of the foodborne disease causing microorganisms are mesophiles, I have already mentioned you. The general rule of thumb is to store perishable foods below 40 or above 140 degree Fahrenheit to suppress the growth of microorganisms. In the temperature range where both mesophilic and psychrotrophic organisms grow, the psychrotrophs grow more rapidly, causing spoilage and at the same time frequently interfering with the growth of foodborne microorganisms. Actually, if both mesophilic and psychrotrophic organisms grow, the temperature window, which is nothing but 41 to 90 degree, that particular temperature condition is favorable for both mesophilic as well as psychrotrophic, but the psychrotrophics grow more rapidly compared to mesophilic, and it causes the spoilage at the same time frequently interfering with the other foodborne disease organisms. The rate of growth always increases rapidly as the temperature is raised. So, this is just a comparison where food poisoning microorganisms versus psychrophilic microorganisms. So, in the temperature range of 50 to 98 degree Fahrenheit, so your food poisoning microorganisms will have rapid growth rate, the same with psychrophilic microorganisms, but the temperature range between 38 and 50, food poisoning microorganisms shows slow growth of some type, and still you will find rapid growth for the psychrophilic. So, psychrophilic, the temperature range is 38 to 98 degree almost. So, your food poisoning microorganisms found no growth around 14 to 38 degree or 35 degree Fahrenheit, which found no growth, and if we see psychrophilic microorganisms, they found slow growth rate, but below 14 degree Fahrenheit, no growth of both, the slow death happens. So, this is the zone of slow spoilage without any danger to health. It is just a comparison how the temperature window affects various microorganisms. So, this is another example, growth of bacteria on the chicken. So, if you see the 32 degree Fahrenheit, it finds almost 18 days. So, the growth is around 10 to the power of 8. Okay. So, but if you increase the temperature, then the growth stops within 8 days, and if you further increase almost to 50 degree Fahrenheit, it is around 2 to 3 days, the maximum is 10 to the power of 9. So, the next important factor is water activity. So, water activity is nothing but availability of water to the microorganism. It is only the roughly related to the presence of moisture. So, the GMP regulation is nothing but a good manufacturing practices. Even I told about the previous slides, the equipment, how the contamination happens in the equipment. So, these regulations to be followed, even how to clean the equipment and where to keep the equipment, and the food processes to be kept in which area, and environmental conditions, everything comes from this GMP regulations, that is good manufacturing practices regulations. For low acid canned foods, defined water activity as the vapor pressure of the food product divided by vapor pressure of the pure water under identical conditions of pressure and temperature. So, the regulation defines low acid foods such as foods other than beverages with a finished equilibrium pH value greater than 4.6. Right, low acid food means normally pH, anyway we are going to see pH is also one of the factors affecting. So, pH 7 means, so that is neutral, so less than 7 that is acid, greater than 7 that is alkali condition. So, in the less than 7 acid conditions also there are two categories, one is low acid and high acid. So, the high acid is below 4.6, the low acid food is above 4.6, and the water activity greater than 0.85. So, the regulations defines the low acid food. So, this is the favorable conditions for the Clostridium botulinum to grow. So, that produces the toxins and also the disease is botulinum. So, we are going to see anyway what are all the symptoms and related food spoilage. So, the conditions for low acid food is pH should be greater than 4.6, at the same time water activity should be greater than 0.85. So, if you compare the water activity, bacteria it is high, and then there comes the yeast, and the last one is molds. Molds we have already seen, it requires minimum water as well as the nutrient condition, even with that they can grow. Certain molds and bacteria can grow on fish immersed in saturated salt solution. So, we told that already the yeast and molds can grow at high salt, even high sugar environment, where the active AW is about 0.75, and some molds can grow in foods with the avactor activity of 0.62 to 0.65. At these lower limits, the growth is very slow. If it has to grow in the presence of sugar, that water activity of about 0.1 is itself enough. The stability of intermediate moisture foods, which is nothing but 0.75 to 0.9, which also includes 0.85, such as dried fruits, jams and soft moist pet foods, depends upon the combination of factors. Though we say that water activity of 0.85 is favorable for the bacteria to grow, it also depends upon other factors, which is low pH, the pasteurization, chemical additives, how much, what are all the chemical additives we add, and the impervious packaging. So, it is not only we can say that if you maintain this particular water activity, your spoilage can occur, but it also depends upon other factors. So, the water activity limits for growth of principal foodborne disease organisms. So, the minimal AW is given. For Salmonella species it is 0.945, for Clostridium botulinum it is 0.95, the Clostridium perfringens it is 0.93, Staphylococcus aureus it is 0.86, and Vibrio species it is 0.94. So, this is we told that minimal AW for the growth of microorganisms. So, to toxin to provide, for example, it may also produce the toxins which causes the further diseases. So, that activity may vary little bit from the growth conditions. For example, Staphylococcus, if it can grow at the water activity of 0.86, but it starts producing its toxins at for say 0.92 water activity. So, these both are both may be different. So, and what is the pH and initial population count? What is the effect of pH and initial population count on the food products? Growth of microorganisms on the food products. So, this we have already discussed, what is less than 7, equal to 7 and greater than 7. So, as we already told, many bacteria grow best at about neutral pH. They grow poorly or not at all grow below pH 4. Right, the favorable is 7, but still it can grow in the low acid food, but below 4 the probability is very much low. Yeast and molds therefore predominant in low pH foods where bacteria cannot compete. This we have seen from the first slide onwards, because the bacteria and yeast and molds require the opposite conditions to grow. The lactic acid bacteria can grow in high acid foods and actually produce acid to give us sour milk. So, the lactic acid bacteria, so this we might have seen, if we keep the milk then at a high temperature for particular period, it become a sour milk. So, the pickles, fermented meats and similar products also suffer with lactic acid bacteria. Some strains called Leucaena stock species, which contributes off flavors to orange juice. Orange juice is highly acidic, but the flavors will go if these species are found in that orange juice. And one more thing is actually the GMP suggests that the 12 log reduction we will talk about when we talk about the kinetic parameters. We talk about this also, the thermal death time. So, they call it as a for at particular temperature to reduce the microorganism count from 1 log. Right, how long it requires. So, that is the time, which is nothing but the thermal death time. So, normally the practice what the food microbiologist follow is the 12 log reduction. So, how long, what is at particular temperature, if 12 log reduction happens, to happen 12 log reduction, how long it requires. So, that is the practice they follow, but still if the initial count is high, then there may be a possibility for the microorganisms to be there in the food, right. That is what the initial population count. So, this is major importance to the processor of refrigerated food, the shelf life of which is enhanced by good sanitation. So, a high level of spores also increases the possibility that few will survive to spoil the heat processed products. That is what I told. So, even though the best practices says 12 log reduction at the particular temperature, but if we have high level of spores, so it is nothing but a probability, right. So, we cannot kill all the spores, it is 90% or 99.9% only, even that 0.1 or 0.001% can cause the spoilage. So, high level of spores increases the possibility that few of them will survive even after it is thermally processed. So, these are all pH values of selected foods. So, the pH values about 2.3 range, so lemon juice and cranberry sauce, and 3 about berries, peaches, orange juice, apricots, and 4 around cabbage red, pears, tomatoes, and 4.6 this is what we told low acid food. So, this is the favorable conditions for Clostridium botulinum. So, this can happen in meats, most of the vegetables, starch based foods and protein heavy foods. So, 5 is nothing but spaghetti in tomato sauce, figs, onions, green beans and potatoes. 6 around tuna, cold fish, beef, pork, evaporated milk, chicken, corn. And 7 is around milk and ripe olive. So, if you know whatever the examples I have taken is for chicken for the bacterial growth, and milk most of them lack the bacteria so affected. So, their favorable conditions are around neutral pH, and the 4.6 is favorable for Clostridium species. The minimal pH minimal for growth of principal foodborne disease organisms is Staphylococcus aureus. So, if you see this is growth reported at but not below this pH. So, below 405 the Staphylococcus aureus cannot grow. The Salmonella it is 4, Clostridium botulinum there are A, B, E, F types, AB types 4.8 and Clostridium botulinum type E it is 5. So, see 0.2. So, the control of pH is very much important, within 0.2 we will get different types. And Clostridium perfringens it is again 5, and Vibrio species it is 4.8, Bacillus species it is 4.9. So, if you see it is mostly between 4.5 to 5. So, these are all principal foodborne disease causing microorganisms. So, the next one is the next factor affecting is oxygen. So, this is essential for growth of some organisms. So, based on that we divide into 3 categories. One is aerobes, anaerobes and microaerophilic. Microaerophilic needs little oxygen, they can grow at little oxygen condition. So, that means aerobes as well as anaerobes. So, strictly aerobes grow only on the food surfaces and cannot grow in the foods stored in cans or in other evacuated hermetically sealed containers. So, normally hermetically sealed containers, when we see canning operations we would be seeing. So, how to remove the air which is contained in the cans. So, because aerobes need oxygen for their growth, they may not grow in the cans, food stored in the cans or other evacuated or hermetically sealed containers. Anaerobes grow only beneath the surface of the food or inside the containers, but when microorganisms are involved, for example, they need little oxygen, mostly are anaerobes, but certain organisms can grow in both the conditions, but in such case if the air is found, their growth will be higher compared to anaerobic conditions. That means they will be seen in the food surfaces. So, then lethal effects of temperature. The most practical and effective means to destroy microorganism is nothing but a thermal processing. So, heat it to higher temperature. So, when you increase the temperature, then the growth of the microorganisms will be decreasing. So, the cell reduction occurs just above the maximal growth temperatures right? Growth temperatures in the sense, for example, it can grow at say 42 degrees Fahrenheit. So, if you increase the temperature above than that, then the cell reduction starts occurring, and the rate of death increases markedly as the temperature is raised. So, when you increase the temperature, the rate of death increases. There are two main thermal processing is involved. One is pasteurization, another one is sterilization. The pasteurization is nothing but a destruction of vegetative cells of microorganisms which are disease causing, which consist of a temperature of 140 degree Fahrenheit for about 30 minutes or 161 degree Fahrenheit for 16 seconds. So, this time temperature combination is also designed by the kinetic processing. So, we will see in the subsequent lectures. Various processing needs various time temperature combinations, and we say low temperature less time, and high temperature less time, and high temperature high time. So, there are many possible ways to come up with the time temperature combination based on the food quality, nutrient quality, and bacterial deactivation. So, that we will see in the next few lectures, but here I would like to mention two examples. If we are maintaining 140 degree Fahrenheit, it is about the processing should be done about 30 minutes, but if it is 161 degree Fahrenheit, it is almost 16 seconds are enough. And yeast molds and the vegetative cells of spoilage bacteria also die at pasteurization temperature. So, remember vegetative cells, if there is as we have seen already, they may form as spores and they can survive pasteurization temperature. When the favorable conditions occurs, they may start converting into vegetative cells. But one thing we should remember is after pasteurization, the storage is important. It should be stored in the refrigerated conditions. The commercial sterility is the destruction and or inhibition of the organisms of public health significance as well as the organisms of non-health significance which could spoil the product. So, here mostly we take care of the yeast molds and vegetative cells, but sterilization is the higher order thermal treatment. So, here we destruct as well as inhibit the organisms of public health significance which both disease causing, otherwise the spoilage organisms of the food. So, the sterilized products which does not require the further storage conditions, it can survive in the room temperature about particular time period, maybe 6 months to 1 year. The low acid food requires temperature above 212 degree Fahrenheit. Canis process certain canned foods at 240 to 250 degree Fahrenheit, it is about 121 degree centigrade. For a considerable length of time, usually 15 to 20 minutes, sometimes an hour or more, this an hour or more. So, if you reduce the temperature, the time temperature combination, there are many. So, normally the low acid food requires 121 degree centigrade sterilization temperature for about 15 to 20 minutes. As I mentioned you already, there is DT, that is nothing but a thermal death time. So, which is nothing but a logarithmic numbers of survivors is plotted against the length of the time. So, this is time versus log reduction of your microorganism. So, for example, 1, 10, 100, 1000 kind of thing. So, 1 log reduction. So, how much time it requires at constant temperature. So, temperature is constant here. Subjected to a given temperature, the results is usually a straight line we normally get. So, this to this. So, this is 1 log reduction from 100 to 10 to reduce that how much time it requires. Actually, we say that it is a straight line, but there are exceptions where we will not get this as a straight line. The slope of this line becomes steeper as the temperature is increased. For example, if it is this is say around 40 degrees centigrade. So, if I increase 50, then my line becomes this, because you require very less time, that I already mentioned. So, if you increase the temperature, you would require less time to kill the same population. required to kill a population at higher temperature. It also takes longer to kill a high population of organisms. For example, so here we put 100, so if I go for 1000 to 10, right, so it requires longer time because my initial starts with 100, then I may require 15 minutes. Initially itself, if I have 10 to the power of 3 number of spores, then it requires more time. The rate of thermal destruction is greater in foods with high water activity than in those with low water activity, the rate of thermal destruction. So, one example is the recommended pasteurization process to destroy Salmonella species in liquid egg albumin prior to freezing. Before freezing, they do pasteurization. So, that is if we maintain 60 degree for 3.5 minutes. So, if it is liquid egg, whereas that for dried egg albumin, it is the say 140 degree or 60 degree centigrade to 158 or 70 degree for several days, right. So, that means if we have high water activity, the rate of thermal destruction is higher compared to low water activity. So, it can be, if it is a liquid egg, it is 3.5 minutes, but if it is a dried egg albumin, it takes several days. So, the temperature also have to be bit higher. The presence or absence of organic matter, oil or fat, pH, strain of organisms, quality of available nutrients, age of the culture. So, these all also affect the time temperature combination. For example, so not only I need to see how much log reduction I will be able to get in a particular time temperature combination. So, if I heat that particular temperature that long hours, so what happens to my nutrients in the food, and what is the pH alteration during that time temperature combination, what is the strain of microorganisms I have, what is the age of culture. So, everything else is also matter when we fix the time temperature combination. And one more thing is like another thing what we think is like we always talk about the higher temperature. So, what happens if I store it in a chilling, right, because I already told the pasteurization, after pasteurization we need to preserve it in the refrigeration temperature. So, what happens during chilling? Chilling to temperatures below the growth range but above freezing stops the reproduction, but kills few cells except for extremely sensitive organisms such as vegetative cells of Clostridium perfringens. So, it stops reproduction, but kills few cells only, but there are some certain sensitive organisms which still survives even at chilling temperatures. I already told, though we give example, but one thing is the probability of the spores present in the food, and also there are many factors which affects, and sometimes the closely related species also will have different effect in the particular temperature. The freeze kills part of the microbial population within few hours, and storage continues to be lethal at much slower rate, which depends upon the nature of the fruit. For example, rapid drop in aerobic plate count occurred in orange juice. So, if I keep orange juice in the chilling conditions, then aerobic plate count we are going to see, this is nothing but a qualitative count of the microorganisms. So, there is a drop in aerobic plate count in the orange juice. So, actually bacterial spores dry very slowly in the chilling temperature, if at all during freezing and frozen storage. For example, vegetative cells of Clostridium perfringens generally all die, but the spores survive. I already told, vegetative cells may die here, but spores may survive even at the chilling temperature. And Staphylococcus aureus and related organisms still survive well in the chilling temperature range. Actually, freezing is not a dependable means to destroy microorganisms, since some cells of the original population almost always survive. So, the freezing comes like before freezing, normally we do either of the thermal processing, pasteurization or sterilization, mostly pasteurization. So, as I already told, even after pasteurization there are many factors which affects the survival of the microorganisms after the thermal processing as well. So freezing is not a favorable condition to destroy microorganisms. It may be used for storage, further storage after the particular thermal processing is employed. So what is this aerobic plate count? So this is aerobic plate count means it is a fraction of the bacterial flora that is able to grow as a visible colonies under the arbitrary test conditions provided in the time period allowed. So particular time period, how much visible colonies I am able to see at particular test conditions. So if the test conditions are changed, then there may be you will get the other number of counts. So the high APC, high aerobic plate count reveals, so it is a one of the measuring technique. Failure of sorting, trimming, washing, destroying operations to remove or destroy bacteria from raw ingredients adequately. As I told, for example, if we take canning operations, so before that they do some of the pre-treatment. One of the pre-treatment is blanching, right. So although it was done to inactivate the enzymes, sometimes it also kills certain microorganisms. So if we still find after those operations, if we still find APC count high, then that means that was not done properly. Though if it is meant to reduce the microorganism count, then it should not give high APC. And inadequate heat processing. Sometimes what happens, though the time temperature combinations is not adequate enough to reduce the microorganisms to the particular log level, and insanitary equipment particularly near at the end of the process, I already told, though after processing is done, even GMP regulations suggest that near the wall also you need to maintain the sterilization temperature or pasteurization temperature. So there is a possibility if the temperature is lower than the particular processing temperature, then there may be a possibility of the contamination. And the food has reached or is approaching the end of its refrigerated shelf life, and sometimes what happens is they say that even it should be used at the refrigerated storage, but the condition is 3 days or 4 days, right. So at that time also we need to probably check in the refrigerated condition also what is the time period or what is the shelf life of the refrigerated product. The food has been sorted at or stored at or above room temperature for too long. So for example, if we see, if something says that if we go for shop and buy some refrigerated food products, if they say it should be kept at refrigerated temperature, we come home and we keep it some 6 hours in the normal temperature, then we remember and keep it in refrigerator. So that storage in the room temperature at 6 hours also counts, also affects on the some of the microorganisms which grow on the food. The food is at least partly decomposed in the sense, even though it is already spoiled, but we store it in refrigeration, that will not improve anything. The refrigeration is nothing but we can restrict the growth of microorganisms. If already there, it cannot do anything about it. So one is coliform bacteria. So this available in all the dairy products. So if the conditions are not maintained properly, then the spoilage occurs due to these bacteria. One thing I would like to mention here is the high AP, whatever the points we discussed, it also lies with the manufacturers. For example, if it has to be stored under refrigerated conditions, it should be legibly written in the cartoon, I mean the label, and also use by date is very much important, because one of the thing what we discussed is and end of its refrigerator shelf life. Even in the refrigeration conditions, it cannot be stored almost 1 year or 2 years, right. There is a use by date for that, even for refrigerated products. The next one is food poisoning. Human illness caused by food-borne microorganisms are popularly referred as a food poisoning. Apart from illness due to food allergy or food sensitivity, we have this, right. For example, I am allergic towards high fat food, or I am allergic to high oily food. So apart from these two, they may be divided into two major classes. One is food infection, another one is food intoxication. The food infection results when foods are contaminated with pathogenic, invasive and food poisoning bacteria or eaten, right. The bacteria itself we had it, and these bacteria then proliferate in the human body, mostly intestine, and eventually cause illness. So, I have eaten the bacteria itself which causes the disease. The second one is food intoxication, which happens, for example, the microorganisms grows in the food, and we have discussed, right, certain pH after there is a toxins are produced by the microorganisms. So, the toxins were produced by the microorganisms. So, I have eaten those toxic substances, right. So, the second one is the ingestion of preformed toxic substance which accumulate during the growth of certain bacterial types in food. So, that is food intoxication. The earlier one is food infection. The period of time between the consumption of contaminated foods and the first appearance of my illness is nothing but a incubation period. For example, morning I have eaten, after 2 hours I started vomiting. So, the in between time is nothing but a incubation period. It ranges from less than 1 hour to more than 3 days, depend upon the causative organisms or the toxic product. So, certain things we discuss here. As I told, this is botulism, the disease. The etiologic agent which causes is a Clostridium botulinum. So, there are A, B, E, F type toxins. So, usually the incubation period is 1 or 2 days, range 12 hours to more than 1 week. So, the symptoms are swelling, double vision, difficulty in speech, occasionally nausea, vomiting and diarrhea. In early stages, constipation and subnormal temperature, respiration becomes difficult, often followed by death from paralysis of muscles of respiration. So, this is a dangerous one. And staphylococcal food poisoning, so the caused by staphylococcal enterotoxin. So, the incubation period is 1 to 6 hours, again nausea, vomiting, cramps and temperature subnormal during acute attack, may be elevated later, rapid recovery usually within a day. Another is salmonellosis. I already told it is spread by cockroaches, flies, and specific infection by Salmonella species, average about 18 hours, range from 7 to 17 hours. So, again abdominal pain, diarrhea, fever, frequent vomiting, then duration of illness is 1 day to 1 week. Then Shigella is bacillary dysentery. So, this is caused by Shigella species, usually 24 to 48 hours. Again here abnormal cramps, fever, chills, diarrhea and headache, nausea, dehydration, these are all symptoms. And Escherichia coli, I already told, coli bacteria. This is Escherichia coli serotypes associated with infant and adult infections. So, usually 10 to 12 hours, headache, fever and diarrhea, vomiting, duration is a few days, it will be remained. Clostridium perfringens food poisoning. So, the agent is Clostridium perfringens, usually 10 to 12 hours. So, abdominal cramps, vomiting is very rare, nausea is another symptom. Meat and poultry products usually involved this kind of bacteria, rapid recovery is possible. Then Vibrio species, it is again virus, usually 12 to 14 hours, abdominal pain and watery diarrhea, usually nausea and vomiting, mild fever also. So, the recovery period is around 2 to 5 days. Then the food spoilage, actually the problem facing by the food industry is spoilage by the bacteria. Though I have already told that thermal processing applied in two folds. One is spoilage bacteria to control growth of spoilage bacteria, another one is disease causing bacteria, right. So, the spoilage is of every food industry concern, which is not hazardous to health. The chilling slows spoilage, proper freezing, drying, canning and pickling arrest it completely. Chilling I already told. So, we need to employ a thermal processing and further the growth is controlled by chilling, and proper freezing, drying, canning also do it completely to arrest the food spoilage microorganisms. Chilled foods must be transported to the customer before spoilage microorganisms make them unfit for consumption. I already told, if some food product should be kept at refrigerated condition, so it should be transferred immediately. So in between the transport and storage at maximum time without the chilling temperature also makes the food to spoil. Refrigerated foods products are convenient to use, have a close to fresh image. As we already told, the refrigerated conditions are maintained for safety, but it is most of the time to maintain the product quality. So it gives a close to fresh look. The optimum storage temperature is 33° Fahrenheit or as close to freezing as possible. However, most refrigerated cases are holding near 45 or even 45° Fahrenheit. There are three things we need to remember even at refrigerated foods. First of all, we have to assume pathogenic organisms are always present in the food product. The second one is refrigeration temperatures may slow or prevent the replication of most of the pathogenic microorganisms, but still some will continue to multiply. Example is psychrotrophs. We have already seen how even at refrigerated temperature. Thirdly, manufacturers should expect some temperature abuse of the foods during storage and distribution. This includes handling at the customer level as well. So manufacturers always need to take care. For example, so from the shop to home or wherever from the place, and there is a transportation and storage also there. So they should take care of that temperature level before it reaches to consumer. Canned foods, the shelf life of canned foods results from the destruction of microorganisms capable of growth within the container during normal handling and storage. They follow GMP regulations for low acid food, that is I already told, low acid food the condition is pH is 4.6 above and water activity is 0.85. Most of this condition is most favorable for Clostridium botulinum. So they have to follow GMP regulations and reduce the spore level in the food. As a general rule, food with a high spore level requires more retard time or temperature in the same similar operations. We have already told, if we maintain the same, for example, there are two cases, one with high spore and another with low spore. Low spore requires low time, low temperature compared to higher one. Assuming same, the incident of spoilage will be higher for canned food with the high initial spore level. The next one is the follow good sanitation and good container handling techniques. So everything should follow GMP regulations. And cooling as well as post cooling period. The important thing is, even though we employ pasteurization or sterilization at a high temperature, immediately the product should be cooled to normal room temperature, because it reduces the possibility of growth of the thermophiles organisms, because the thermophiles survive at higher temperature. It should be cooled to normal room temperature to avoid the growth of thermophiles. So, and also maintain good seams and cans, because the airtight container should maintain the perfect sealing to control the growth of the microorganisms. Okay, so I have shared one particular table which is taken from the prevention of microbial and parasitic hazards associated with processed foods, a guide for the food processor, and the pH value as well as the water activity value were taken from some of the references, and the rest are the books and references which are used in the particular lecture. So that's all for today. So tomorrow we will see about next lecture. Thank you.